Mhm. Welcome everyone to this week's edition of the Extracellular Vesicle Club or EV Club. We're really happy to have you today, and we're going to have a very nice presentation that was just recently published in Nature Communications. So some of you may have been at the ISEV annual meeting recently that was held in Vienna, Austria. And if you were there, or if you've been following along on social media, you know that therapeutics has been a very big theme at this year's meeting. Probably the largest number of presentations on EV therapeutics that has ever been gathered in one place before. And this week on EV Club, we're going to be continuing that theme with Gi-Hoon Nam, who's representing the Korean Institute of Science and Technology, KIST, as well as a bio company that's known as Shift Bio. So we're going to hear a little bit more about that and about some of the exciting work that Gi-Hoon and his colleagues are doing. So Gi-Hoon, welcome. Thank you for presenting today, and
Mhm.
I look forward to what you have to share with us.
Hello, everyone. My name is Gi-Hoon. Thank you for being here today, and thank you, Ken, for introducing me. I really appreciate this opportunity to present our science and technologies at this EV Club. I am big interested in advanced drug delivery system using natural nanoparticle, especially extracellular vesicles. Actually, I am feeling under the weather right now, but I will do my best to present my science and technologies. This is my disclosure. I have two affiliations. I serve as co-founder and CEO of Shift Bio, and I am an assistant professor at Korea University College of Medicine. The goal of this presentation is to introduce our recent publication on high purity, high efficient, large-scale manufacturing process of EVs and engineered mesenchymal stem cell EV-based in vivo macrophage reprogramming strategies for treating inflammatory conditions. Okay, to start off, I want to ask you one question. Why extracellular vesicles? Why are we doing extracellular vesicles? Extracellular vesicles or exosomes were quite buzz about 5 years ago. Not just for me, but for the entire industry and scientific communities. But today, those enthusiasm has cooled down due to the several reasons. Many people were still skeptical of a large-scale manufacturing process of EVs for clinical translation. And many people still and also believe that exosome leading company Kodiak bankruptcy is a clear evidence that extracellular vesicles is useless as new drug development. But for me, those perspectives are too boring and don't leave much of impact anymore. We all know that history repeats itself. In the 1990s, people didn't believe in antibody drug ability. In 2000, antibody drug conjugate were considered as bundle of toxicity. In 2010, cell therapy were thought to be impossible due to their heterogeneity. In 2020, mRNA leading company almost went to bankruptcy before COVID-19. However, we all know that these modalities turn out to be game changers in new drug development market. History echoes itself, and the time for extracellular vesicles is just around the corner. The unchanging truth is that extracellular vesicle is the promising modality and innovative drug delivery system. So the question we must ask ourselves again is, why we need to use extracellular vesicles? What can we serve using extracellular vesicles? Why extracellular vesicles? To date, current drug delivery systems have transformed many promising cargoes into meaningful medicines so far. However, these current drug delivery systems have big limitation in safety, immunogenicity, targeted delivery, tissue penetration, and endolysosomal escape. We believe that natural nanoparticle, including extracellular vesicles or non-vesicular extracellular particle like supermere, can effectively solve these problems. These nanoparticles offer excellent biocompatibility and superior drug delivery efficiency. Currently, over 183 clinical trials are using natural nanoparticles worldwide. And one example using mesenchymal stem cell-derived EV has advanced to phase 3 in the FDA. This is a clear evidence that natural nanoparticles, including extracellular vesicles, are practical for medical use. This is not for medicine. This is a medicine that's arriving soon. Despite this great potential of natural nanoparticles, difficulties in complex engineering and resource-intensive manufacturing have been regarded as major bottlenecks of NMP realization from science to patients. To address these challenges, we have created our natural drug delivery platform for engineering and manufacturing natural nanoparticles, including extracellular vesicles. Let me explain our engineering approach first. By uncovering how specific proteins were naturally sorted onto extracellular vesicles during their biogenesis, we have created our own specialized EV engineering scaffold. We are calling this the Natural Nanoparticle Sorting Motif, NSM. This NSM is a very short transmembrane domain. Our own transmembrane domain can effectively transport into the bilayer of extracellular vesicles through the interaction with specific tetraspanines. So this is a platform technology that engineer extracellular vesicles to effectively load macromolecules inner side of EV and to express the targeting moieties on the surface of EV. This technology has 3 distinctive advantages. First, high engineering efficiency compared to current engineering scaffolds such as PTGF-RNA or tetraspanines. Second, multiplex cargo loading. This means that we can load several cargos into and onto single nanoparticle. Third, universal cell application working both in HEC293 cells, genetically easily engineering cells, and primary cells like mesenchymal stem cells. After developing our NSM scaffold, we faced an important questions. As you know, traditional EV engineering requires significant cost and labor. So our question is, how can we streamline and speed up this EV engineering process? The solution is an AI-based in silico modeling system for EV engineering. We have used the Dassault system in silico modeling system, and we incorporate this in silico system into our NSM scaffold. And then we finally created NMP design AI platform technologies. This platform operates in 2 main steps. The first one is this can identify the structure of NSM cargo fusion protein using modeler system. Second, we analyze the protein-protein interaction between NSM, our EV engineering scaffold, and specific tetraspanines, which is crucial for EV engineering. So we can predict the success of EV engineering before experiments, biological experiments, and we can significantly reduce the timeline to create our advanced drug delivery system using extracellular vesicles. let's move on to our CMC part. We recently published our manufacturing platform technologies in Nature Communication, where you can find more detailed information. But today, I want to briefly introduce how we can overcome the bottlenecks of EV engineering and EV manufacturing for large scale and high purity. We optimize upstream process using 3D bioreactors with microcarriers to produce high-yield engineered extracellular vesicles. We demonstrate that 3D-based culture system using bioreactor can increase EV production yield almost tenfold than compared to conventional 2D-based culture system. We completed our comprehensive downstream process to purify engineered EV from the media using depth filtration, tangential flow filtration, and multimodal chromatography. This comprehensive downstream process can effectively purify and recover engineered EV from the media, and our result shows over 95% purity and 50% recovery rates of EV particle numbers. These achievements are really meaningful in this industry because we can significantly decrease the cost for EV manufacturing cost. So based on our engineering and manufacturing platform, we are confident that we can overcome the major bottleneck of EV therapeutics like manufacturing and complex engineering. Based on these platform technologies, we have diverse pipelines for targeted delivery or oligonucleotide delivery and brain-targeted delivery. But today, I will show one example. This is about in vivo macrophage reprogramming strategies using engineered mesenchymal stem cell-derived extracellular vesicles. When developing therapeutics for inflammatory conditions, we focused on two critical questions. The first one is, how can we effectively target inflammatory tissues? The key challenge here is identifying the right target and right delivery tool that reduce toxicity while reach the specific area. The second question is, how can we truly resolve inflammation? Is it enough to target a single inflammatory cytokine or receptor, or do we need to use comprehensive approach? These two questions have guided the development of our in vivo macrophage reprogramming strategies using extracellular vesicles for inflammatory conditions. CD47, known to be overexpressed in cancer, have been discovered by independent research institutes to be also overexpressed in diverse inflammatory conditions, including atherosclerosis, MASH, viral infection, and fibrotic condition and autoimmune diseases. Therefore, we believe that this CD47 is an ideal delivery target for inflammatory and autoimmune diseases. By leveraging our NSM platform technologies, engineering platform technologies, we highly express SERP-alpha protein on the surface of EV, which is the ligand for CD47. Second, our nanoparticles were generated from genetically transduced human bone marrow-derived mesenchymal stem cells. So this means that these nanoparticles can carry stem cell cargo very effectively. So this nanoparticle has dual mode of actions. The first one is this nanoparticle can target CD47 overexpressed inflamed tissues because of SERP-alpha expression. And they can also deliver regenerative cargo originated from the mesenchymal stem cells. And we found that this nanoparticle showed a therapeutic efficacy, and this efficacy is dependent on in vivo macrophage reprogramming. Let me share our probable concept data to demonstrate this. We first identified and found that our genetic engineering of mesenchymal stem cell using our NSM platform to express SIRPα protein on the surface could not change the fundamental properties of mesenchymal stem cells. And we also found that stem cell-derived SIRPα retain the same regenerative cargoes, proteomic cargoes, to unmodified mesenchymal stem cell-derived EV. This means that our platform can fully leverage the therapeutic efficacy of stem cells even when they are engineered. Through the comprehensive characterization test guided by IACV, and we found that our SLP-NMP produced from previous I showed that our manufacturing platform, we produced this SLP-NMP, and we found that this NMP showed the batch-to-batch consistent physicochemical properties. And this means that this manufacturing can be applied to consistent EB productions. For the purpose of proof of concept, we used multiple acute liver injury models. We first found that this intravenous injected therapy NMP can preferentially accumulate in inflamed liver tissues compared to normal tissues. And we also checked the CD47 level on these tissues. These inflamed liver tissues highly express CD47, especially on necroptic hepatocyte and pathological macrophages. And this accumulation is dependent on CD47, demonstrated by pre-blocking CD47 antibody experiments. And we also show that in the cellular level, this SIRPα can target macrophages and necroptic hepatocytes. Regarding the dual mode of action of these nanoparticles, we first found that the first one is this SIRPα can effectively target and block the CD47 on necroptic hepatocytes and macrophages. And by blocking SIRPα CD47 interaction, this nanoparticle can enhance the macrophage efferocytosis against the pathological cells, which are the core of inflammation aggravations. And this means that this nanoparticle can remove pathological lesions very effectively. The second mechanism is that, this SIRPα originated from mesenchymal stem cells. We found that a mesenchymal stem cell-derived SIRPα has the several regenerative cargoes, identified several regenerative cargoes, compared to non-stem cell HEC293 cell-derived SIRPα. And we also confirmed that non-stem cell-derived HEC293 cell-derived SIRPα is not effective in the treatment of acute liver injury, but our mesenchymal stem cell-based SIRPα is really effective in the treatment of acute liver injury. And also, we isolated macrophages from the inflamed liver tissues, and we did the transcriptomics. And we found that mesenchymal stem cell-derived SIRPα can change and reprogram gene signatures of macrophages to enhance regeneration and tissue repair capabilities of these macrophages, while the HEC293 cell-derived SIRPα did not. Based on this dual mode of action of these nanoparticles, we checked the therapeutic efficacy of SIRPα across the multiple acute liver injury models, and we found that this therapeutic efficacy is really promising and shows the superiorities over current medicine, that is the N-acetylcysteine, which is the only therapeutic option for acute liver failure patients. Most importantly, these pipelines in vivo macrophage reprogram strategies is not limited to a single disease like severe liver injury, but also expanded to diverse inflammatory conditions, including idiopathic pulmonary fibrosis, MASH, acute kidney injury, and inflammatory bowel diseases. We are currently working on this research, and these are unpublished data, but I can disclose the data later. So I will share a brief data on these indications. Yeah, in the case of idiopathic pulmonary fibrosis, intravenous injected the same therapy NMP can effectively target fibrotic lung tissues compared to normal lung tissues. And also we found that this therapy NMP can effectively reach pathological macrophages and fibroblasts where CD47 overexpressed. Through the in vivo macrophage reprogramming strategy of this nanoparticle, we found that this nanoparticle produced outstanding antifibrotic efficacy in idiopathic pulmonary fibrosis model. Yeah, move on to mesh therapies. As you know, mesh has the fibrosis, and we first demonstrate the therapeutic efficacy of this nanoparticle in independent liver fibrosis models, and we show that this nanoparticle can enhance the antifibrotic efficacy very efficiently. Through the human spatial transcriptomics data, we identified that macrophages and fibroblasts in human samples highly express the CD47 compared to other cell clusters. And through the same mechanism, this intravenous injected therapy NMP containing mesenchymal stem cell cargo showed remarkable therapeutic efficacy in 2 types of mesh models, and also they showed a promising antifibrotic efficacy in these models. In the case of acute kidney injury, both in mouse data and human data, we identified that macrophages, actually pathological macrophages, highly express the CD47 on their surface in acute kidney injury in kidney tissues. And also we checked the therapeutic efficacy, multiple AKI models, and we found that this nanoparticle can be effective in this condition too. Finally, we recently focused on inflammatory bowel diseases because we found that this therapy NMP, when we injected this NMP through the intravenous injection, and we found that this NMP can really effectively target inflamed intestine tissues compared to unmodified NIV or in normal conditions. And we demonstrate that the therapeutic efficacy of therapy NMP increased with dose escalation, and even this can regenerate damaged intestine very effectively. This result collectively indicate that this in vivo macrophage reprogramming strategy using our SELP NMP engineered mesenchymal stem cell derived EV can be promising option for treating inflammatory conditions. And we are aggressively conducting research on developing this nanoparticle as the next Humira. Long story short, we aim to develop in vivo cell therapy using natural drug delivery platform technologies. And we are confident that these technologies can give a new chance to patients who have no therapeutic options. But still someone say, why natural nanoparticles? We choose the natural nanoparticle to create our medicines, not because they are easy, but because they are hard, because that goal will bring the next paradigm shift in drug delivery systems, because that challenges will make the innovation accessible from science to patients, even to the market. Thank you for listening to my presentation. Thank you.
Well, thanks very much to you, Gi-Hoon, and I'm sorry to hear that you're under the weather, and I really appreciate your giving the presentation nonetheless, and I'm sure that all of our audience members are as well. So I'd just like to remind everyone that if you have not put your question or comment in the chat box, please do so now. I'm currently just toggling the switches here so that you can unmute yourselves, and you can also show your video if you'd like to. And we will get to the questions right away. So I see that we already have a question here from Qingling Fu. So Qingling, if you'd like to just go ahead and have a conversation here with Gi-Hoon, please go ahead.
Okay, thank you, Ken. And It's a very excellent work. I have some question about the
Thank you.
preparation of SEVs. And I'm wondering how about the macro carriers? Because you did not present clearly in your presentation, which company it is from, and how about their characteristics? And the second question is the cells, because you mentioned that the cells have been cultured in free protein fridge, chemical that define the low particulate medium for 5 days. I think it was a little long. Now, how about the cells condition? Whether some cells became apoptosis or something like that. And the third question is, I noticed that.
Ling, sorry. Let me just interrupt there because I'm sure that Gi-Hoon can keep them all in his head, but I'm getting older and I can't remember all the questions at once. So let's just let Gi-Hoon answer the first two, and then we'll get to the rest of them. Go ahead, Gi-Hoon.
Yeah, I just put the comment in the chat box. Yeah.
Oh, sure. Yeah.
You can read the question in the chat box.
I got it. I got your points and your questions. Yeah, very good questions.
Okay.
About the microcarriers, I can't disclose the company name and the catalog numbers right now, but we use the highly already proved the in the clinical trials. We use the common microcarriers to culture the adherent cells, because mesenchymal stem cells are adherent cells. So we have to use microcarrier to incorporate these cells into bioreactors. So we use the commonly used microcarriers in clinical trials. And the second question is, very good questions. Yeah, I think the 5 days is a little long for culture of primary cells, but we found that until reaching the passages 7 of mesenchymal stem cell, that is okay, determined by several parameters of cell characterization, like differentiation properties or secretion of a bunch of tons of cytokines and proliferation rate. So we aggressively checked this process can sustain the properties of mesenchymal stem cell. We found that this timeline is okay with engineered mesenchymal stem cell, because we use the very early passage of mesenchymal stem cell. Actually, this is the GMP version from the Rooster Bio, and we checked the stability and characteristics of mesenchymal stem cell during our process. So 5 days is okay in our view. And second question is, 50% recovery rates. Sometimes I want to emphasize this figure, 50%, 50%. This means that we can only discard 50% of EV to get the high purity engineered extracellular vesicles. But this is the industry common sense. In the case of HEC293 cells, the recovery rate, I think, is around 5%. That means 95% of EV can be removed through the downstream process to get the clinical accessible EVs, to remove the impurities like double-stranded DNA or albumin or other types of nucleic acid. So I think 50% is really a significant figure in this industry, because we can reduce the cost of EV therapeutics and production cost around tenfold compared to current manufacturing process. And number 4 is, we use the, I think, hollow fibers, thin junction and hollow filtration, commonly used in EV field. And pore size is a little know-how. So, but we already disclosed we can the information we can disclose, we already put in our manuscript in papers, so you can find more detailed information. And the 5 question is, yeah. Right. Yeah. We use the bone marrow-derived mesenchymal stem cell, and the limitation of passage number is around 10. But we found that until 7, passage 7, the mesenchymal stem cell properties are okay. But the reason why we are choosing human bone marrow-derived mesenchymal stem cell is that there are so many evidence using these cell sources for clinical trials of exosome therapeutics and extracellular vascular therapeutics. So yeah, because we focus on platform technologies, so we want to use the cell source which have a lot of previous results in clinical trials. Yeah, thank you.
Thank you very much.
Mm-hmm.
So Qingling, I was wondering if you could elaborate. You mentioned that you felt that the BM-MSCs are not a good candidate for engineering. And
Yes,
is that
because
solely because of the passage numbers in your view?
Yeah, because the bone marrow derived MSCs had about the limitation proliferation about less than 10 passages. I think that the common passages used for the bone marrow MSC is about the 4 to 6 passage. Because for the engineered MSCs, I'm wondering how about the which passage for you to do the transfection, passage 0 or passage 2?
Passage 2, yeah, right. Passage 2.
Yeah, because after the transfection progress, and then you will go to the 4th or 6th passage, right? And you use the 7. You use the 7 passages. So it's not a good candidate for the engineered SEVs using the MSCs, right?
Right. Very good. Yeah.
Good. All right. Well, thanks for the questions, Qingling. Very, very helpful. So I'm not seeing other questions in the chat box at the moment, but if anybody does have questions, please go ahead. You can also raise your hand if you want to. So Gi-Hoon, I was really encouraged by not just the paper that you presented, but also the the new data that you showed as well with some of the different disease models. Now, how about delivering these EVs to targets other than the liver? Are you exploring ways to get them into other solid tissues as well?
Yeah, very good question, Ken. And yeah, as you know, in normal mice, this is the mice, but in normal mice, generally, extracellular vesicles preferentially accumulate in the liver and kidney. But in the inflammation condition, if we express the therapy protein on the EB surface, this therapy EB can accumulate in inflammatory tissue very effectively. So in the case of inflammatory bowel disease, we found that the accumulation rate of this extracellular vesicle into the inflamed intestinal tissues is comparable to liver accumulation. This is really outstanding findings. So I think that EB has the great advantages for extrahepatic delivery, but we have to find out the right target and right engineering strategies. So because conventional other nanoparticles can go into the many cells because they shows the high endocytosis rates, but we believe that extracellular vesicles can evade unexpected endocytosis and uptake through the surface engineering. So we believe that CD47 is also ideal target for extrahepatic delivery and targeted delivery. So we already checked the feasibility of this nanoparticle in other types of conditions.
Fantastic, thank you. We now have a question from Hui Chun. Please go ahead.
Hi, thank you. Oh, it was a fantastic talk. I'm just curious about your chronic model because you only show the result. Because how long can your EV remain active in vivo? So for those long-term ones, obviously you don't just give one dose.
We use the two mesh models, and first one is in two models we use the multiple dosing strategies of these nanoparticles. So we injected these nanoparticles via intravenous route 2 times a week for 4 weeks. So total 8 times, and we checked the immunogenicity and toxicity of these multiple dosing schedules of this nanoparticle in mouse models. But these extracellular vesicles, is generally exceptional safe. So we choose this multiple dosing schedule, but we have to optimize the effective therapeutic schedule for chronic conditions like MASH and idiopathic pulmonary fibrosis and inflammatory bowel diseases. We have to do more research on these things, and we check the immunogenicity too.
For your IPF, did you see the EV get into neutrophil as well, in addition to macrophages?
Wow. Very good question. So actually, we found that extracellular vesicle can also enter neutrophil, but the important thing is that this is the relative things, but we found that our SERP EV can preferentially enter and reach the pathological macrophages highly expressed CD47.
So next question probably is more about perspective of how when you need to translate to patients. I would assume it will be a huge quantity. How you can meet the clinical need?
Could you rephrase your question? Sorry.
For when you give the EVs to the patients, the quantity you require is much larger than an animal experiment. And then so for bone marrow MSCs, they grow really slow. And then so even for past that number, like how you meet the clinical need when you look at the patients.
Very
you don't look at just the 10 of them. It's actually could be tens of thousands.
Very good questions. But I want to say that your question was really important for us 4 years ago, because we were skeptical of using mesenchymal stem cell primary cells because of your questions and your reasons. But we found that one cell can produce 1,000 and 10,000 extracellular vesicles. This means that if we can optimize the manufacturing process to get sufficient EV from the cell media, I think this is feasible approach, even we used primary cells. In the case of our SERP EV, we got the orphan drug designation from FDA last year, but we showed that this 200 liter, 200 is not large scale for antibody, but 200 liter scale can produce 8,000 dose for acute liver failure patients determined by our calculation of dosing. So this means that this scale can be enough for early clinical trial, because the reason why this approach is possible because we developed very high recovery rate manufacturing process, and we found that this can be enough for making transformative medicines to give patients using these dosing strategies.
I don't know whether this is a commercial secret. If it is, you don't need to answer. Do you use anything to actually stimulate the cells to produce more EV than what natural status? If it's a commercial secret,
yeah,
you don't
yeah,
need to answer. I
yeah,
just
yeah.
curious.
Yeah, we don't use any types of priming factors in culture system, but we used several types of reagent to enhance EV production yield, and yeah, actually in EV collection period. I think, yeah, there are so many companies to enhance this EV production yield, and they showcased several reagents, but we used the RoosterBio reagent because RoosterBio is our strategic partnership for this manufacturing process. And they recommended some types of reagents to enhance the EV production yield. And we found that this can also contribute to make high yield of EV in this manufacturing process. Yeah, thank you.
Right. Thank you.
But 3D bioreactor can increase the tenfold compared to 2D culture system. We don't know the mechanism, but I think that stress or stir environment can enhance the EV production yield very effectively too. Yeah.
Thank you.
Yeah. Thank you.
Thanks for the questions. We now have a question from Woo Il Choi. Please go ahead.
Thank you for a great presentation. You used the strategy to deliver EV by targeting CD47. So what is your expected pathway for EV entrance into the cell? For example, endocytosis or fusion, something like that.
Very wonderful questions. And yeah, that is our next research, because we found that SELP-EV can enter the cells through the specific endocytosis mechanism. This is not the traditional nanoparticle endocytosis. This is the clustering independent endocytosis mechanism. And we found that this mechanism may evade the endolysomal pathway like fusion uptake mechanism. So we have to do more research on these points, but yeah, as you mentioned, these points, SERP can target CD47 overexpressed cell, and then they can bind the CD47 and they can enter the cell through the CD47-mediated endocytosis. We already checked the CD47 level on the surface. After SELP-EV treatment, we found that CD47 decreased the cells decrease the CD47 level on their surface. And we also checked whether this SERP-EV can deliver cargo such as oligonucleotide effectively compared to unmodified extracellular vesicles. We found that this SERP-EV can deliver oligonucleotide into the cells very effectively compared to lymphopectamine and unmodified extracellular vesicles. So your question is really great, because the current drug delivery system have to overcome the targeted delivery issues and endolysomal escape. But I think the one example, this is just one example, but SERP-based targeted delivery is promising for these points. Yeah, thank you.
Thank you so much. Yeah.
I find that very interesting to hear, because of course you can have the CD47 blockade without even any fusion. But you're telling us that in addition to that, you're also able to use these EVs to deliver cargo. So that's very exciting to hear.
Right. Yeah.
Qingling, you have another question about the MSCs. Please go ahead.
Yes, I have another question. And I'm wondering, do you check the chromosomes about the engineered MSCs? Generally, the transfection for the cells would mostly draw the change of karyotypes.
Yeah. Frankly speaking, we didn't check the chromosome change of after the transduction, but we have to check it because I think FDA required this data package. So, but I think that this is my assumption and my thoughts. There is no significant change during this transduction, but we have to check it. Yeah, your question is really important for enabling FDA approval. Yeah, right.
But for EVs, because EV have some advantages for the cells, because because the EVs are not the cells, and the EV have no the chromosomes, but some EVs more have some mitochondrials. Yeah, mitochondria may have some for the gene, something like that. Yeah.
Yeah. Right. So yeah. So sometimes I think that we use not cells, we use just extracellular vesicles from the cells. So, you know, I also run my company, but we have to streamline the characterization test of our products, including cells. But yeah, I think that your question is really crucial for making the FDA package. But sometimes I believe that I'm not sure the reason why we have to check the chromosome about the MSC, because MSC can have problem like tumorigenicity or something else. So, but I think the extracellular vesicle is really safe in these issues, and we found that our mesenchymal stem cell derived EV almost are removed from nucleic acid contamination. So yeah, but we have to check it in more detail in the future.
Okay, thank you. Very excellent work. Thank you, Ken.
Thank you. And thanks to everybody who has joined this session. We really appreciate you being in the audience and also asking some questions here of Gi-Hoon. But most of all, thank you, Gi-Hoon. Thank you and all of your coworkers. I just want to wish you and your colleagues at KIST and also at Shift Bio, Rooster Bio as well. Thanks for this wonderful work, and we wish you the best as you move this towards the clinics. So very exciting to see.
Thank you very much, Ken.
All right. Well, thanks everybody, and hope you have a good rest of your day, evening, wherever you may be. And look forward to seeing you on another EV Club coming up soon. Thank you.
Yeah.
Bye now.
Thank you. Have a nice one. Bye.